next section, we're going to start to talk about proteins. When you think about proteins, you probably think meat, you think muscles. There's a lot more to proteins than that. And in fact, they are probably the most important biological molecules aside from nucleic acids. Proteins are polymers of amino acids. So in proteins, the monomer is an amino acid. Now remember when I first talked about polymers, I said they were like Legos. Starch is where you build like a tower of Legos out of one kind and color of Lego. Proteins, where you can build a tower, a spaceship, Ferris wheel out of lots of different colors and sizes of amino acids. Okay, so there are 20 different amino acids in nature. Think of that as 20 different Lego blocks. Each of them has the same primary structure and then one functional group that's different. So an amino acid has a carbon in the middle, okay? And on one side it has what we call an amino group, which is a nitrogen with two hydrogens, NH2. On the other side, it has a carboxyl group, a carbon double bound to an O on one side and an OH on the other. And then it's got a hydrogen, and then the fourth bond to the carbon, to our central carbon, is what we call a side chain or a functional group. And this is the thing that's different in each amino acid. So this part, the amino group, the carbon, the hydrogen, the carboxyl group, that's the same in every single amino acid. The side chain, though, is going to be different. And when the side chain changes, it's going to drastically change the function of the amino acid. That's going to make important differences. When we make a protein, the amino group, the carboxyl group, and the side chain are all functional groups. Remember, a functional group is a group of atoms directly involved in a chemical reaction. The amino group and carboxyl group are going to be involved in binding the amino acid monomers together to make a protein. The side chain is going to be involved in other chemical reactions that form the shape of the protein. All right, so when we make a protein, we're going to bind amino acids together. We take the carboxyl group of one amino acid and we make a chemical bond between that and the amino group of another amino acid. Easy peasy, we've got an OH on one and H on another. We do a dehydration reaction. Remember, this is exactly the same reaction we did when we were putting together monomers of sugar. Take the water out, bind the carbon to the nitrogen directly, and we make what we call a peptide bond. So a peptide bond is between two amino acids. This is a term you need to know. Okay? A peptide bond is a bond between two amino acids. When we have just a few amino acids together, we sometimes call the whole molecule a peptide. This would be a dipeptide, okay, two amino acids. Often it's called a polypeptide, especially if there are more than two peptide bonds. It's not a protein unless it's 50 or more amino acids long. So proteins are, by definition, very long molecules. Some of them are hundreds and hundreds and hundreds of amino acids long, maybe thousands. So if it's small, it's a polypeptide. If it's fewer than 50 amino acids, larger than 50 amino acids, it's a protein. Either way, between the amino acids, there is a peptide bond. Okay? And then there could be another peptide bond on this side, another peptide bond on this side, and so you have a big long chain. That chain of amino acids is what we call the primary structure of the protein. And each protein is different in the order of the amino acids, essentially in which Lego blocks we use in which order. Now, you do not need to know the names of all 20 amino acids. You don't need to know the shorthand for them. Don't worry about that. Just know that for each of these 3-letter codes, that's a different amino acid. Okay, so ALA is alanine, MET is methionine, LYS is lysine. Again, you know this. ARG is arginine. The pirate's favorite amino acid. Get it? Arginine. If you change the primary structure of the protein, then you change the way the protein acts, because things that have a different shape function differently. In some cases, the change in a single amino acid can create an entirely new protein. Hemoglobin, remember, that's the molecule that binds to oxygen in the blood. Remember, it's got an iron ion in it. It has 4 iron ions in it, actually. In a disease called sickle cell disease, there is literally one amino acid that's different. And instead of having the normal shape, the protein binds in the wrong shape. Because the hemoglobin binds in the wrong shape, the red blood cell, which is full of hemoglobin molecules, actually takes on the wrong shape. Now, this is important because the smallest blood vessels in your body, called capillaries, are 1 red blood cell wide. And your red blood cells normally go through fine because they're nice and round and squishy. But if they have pointy things on the end, they literally get stuck in the capillaries. And so people who have sickle cell disease run the risk of getting their red blood cells stuck in their capillaries. For reasons that are not very well researched, this happens more often when the body is under oxygen stress, when you're exercising hard, breathing hard, or when you're at high altitude where there's less oxygen in the air. And they're more likely to have a sickle cell incident and lose circulation to part of their body, and they could even die as a result of sickle cell disease. For those of you who are wondering, wow, that sounds like a terrible disease, why hasn't there been more study of it? The answer is because sickle cell disease is most common in sub-Saharan Africa. And most of the people who live in sub-Saharan Africa are Black, and racism is alive and well in medicine, and people didn't want to spend money to study a disease that only affects Black people. For those of you thinking there must be another reason for that, there really isn't. That's really the reason. Anyway, we will come back to talk about sickle cell disease more when we get to genetics, because it's actually a very, very cool genetic story and very interesting from an evolutionary point of view. At this point, we just need to know that this one change in amino acid in the primary sequence, that is the order of the amino acids, one wrong amino acid, you get the wrong shaped cell instead of the normal hemoglobin molecule. All right, now that if we have a primary structure, we're probably going to have a secondary structure as well, and we do. The secondary structure is how the chain folds. Now remember, the amino acids are the same except for their side chains. Those side chains, because they're different, they're going to have different functional groups on them, and they are going to bind differently. So when they bind differently, the two main shapes of binding are a beta pleated sheet and an alpha helix. They named them, they found the helix and then the sheet. So the helix is a twisty, swirly thing, right? That's what a helix is. And then the pleated sheets are literally just folded back and forth. Okay, so those are the two shapes of our secondary structure. So we got the primary is the order of the amino acid. Secondary is the first fold. Then we're going to do a tertiary, or a third level of structure, and that is more complex folding. So the pleated sheets and the helices fold together to take on a 3D structure. Now, for many proteins, the structure stops here. This would be a complete protein, but not always. Some more complex proteins have what we call a quaternary structure, a fourth level structure, in which now the proteins become monomers of a larger protein. So we actually have two levels of monomers here. We have the amino acids, which are the monomers of the protein, which folds into this big protein molecule, which then becomes a monomer of a larger protein molecule. So this is a large protein molecule made out of 3 monomers, 3 sections, and each of these is an individual protein molecule made up of monomers of amino acids. Make sense? Yeah, it's a little confusing, a little complicated, but once you get that there are layers upon layers, there you go. Hemoglobin is a protein made out of 4 monomers of 2 different types. Okay? Not all proteins have a quaternary structure. Some only have the tertiary structure. Now, when proteins lose their shape, they do what we call denature. They can lose when they lose their shape, it's because the chemical bonds holding the folds together start to break down. Those are going to break down before the bonds between the amino acids, the peptide bonds. Okay, so the bonds holding the folds together, these and these and these are going to break first. You still have usually a long strand, the primary structure, but the secondary structure of the protein breaks down. That's denaturing. Denaturing can happen in high heat, sometimes in low heat, in very acidic conditions or very basic conditions. There are a number of different things that can cause a protein to denature. Now, proteins have a lot of very important functions. One of the functions they have is they are enzymes. They help chemical reactions happen. But most enzymes only operate at a certain temperature and a certain pH, which is why maintaining an internal body temperature and a pH level of your blood and internal body fluids is vitally important, because at different pH, your enzymes don't work. Now, think about your stomach, which remember is full of acid, right? Your stomach contains enzymes that work in very acidic conditions. When the digested stuff from your stomach gets dumped into your small intestine, it gets neutralized by a very basic fluid from the pancreas, and those enzymes from the stomach stop working because now they're at the wrong pH, and a new batch of enzymes gets added to break down the food. Cool, right? Now, when we cook food, and the shape and flavor of the proteins changes, that's because the proteins got denatured. Cool, right? Okay, if you don't remember that, just remember, hey girl, you're so hot, you denature my proteins. All right, so let's review our proteins. Proteins are polymers. The monomer in the protein is an amino acid. There are 20 different amino acids in nature. Fewer than 50 amino acids in a molecule, it's called a polypeptide. More than 50, it's called a protein. Different proteins have different combinations and orders of amino acids. That's the primary structure. Different proteins are folded differently, and that folding creates the function. Proteins function really so many things: movement, energy storage, structure, making other chemical reactions happen. That's enzymes. So many things in cells, in our bodies, in plants, depend on the presence and the proper shape and function of proteins. And that's the end of that section.